Shuttle!
As you can see from the MRI results here,
what do you guys see?
Well, doctor, he appears to have an abnormally large liver,
And
that coupled with an estimated large liver size. Doctor, this patient has hepatosplenomegaly. His family's history doesn't say anything about family members having a liver or spleen disorder.
Torres, you've been awfully quiet in the back there. Would you get a complete genetic profile of the both parents and be quick about it?
Niemann-Pick disease. Niemann-Pick disease is an inherited condition caused by abnormal lipid metabolism. Individuals are unable to properly metabolize, break down, transport, and use fatty acids and cholesterol. As a result of the abnormal lipid metabolism in affected individuals, there is accumulation of lipids and cholesterol in the spleen, liver, lungs, bone marrow, and brain. This may result in the enlargement of these organs, which may cause pain to affected individuals. Endosomes are largely responsible for transport of macromolecules. They primarily are sorting organelles where many ligands dissociate from their receptors in the acidic pH of the lumen, and from which many of the receptors recycle to the cell surface. It is also the site of sorting into the transcytotic pathway to late endosomes. Cells acquire cholesterol either by de novo synthesis in the endoplasmic reticulum or by internalization of cholesterol-containing lipoproteins, particularly low-density lipoproteins, or LDLs, via receptor-mediated endocytosis. Niemann-Pick type C affects the processing of cholesterol, both assimilated and generated through lipid biosynthesis. This diagram seeks to explain the mechanism of endosomal transport in a regular individual. As seen here, an LDL particle is first identified by a receptor protein. This is step 1 in receptor-mediated endocytosis. Following this, the LDL is transported by a transport vesicle where it enters an early endosome. Early endosomes are primarily characterized by the presence of a vesicle. This vesicle in the diagram is in the form of a bud. However, it is usually more elongated. The purpose of this vesicle is to transport receptors back to the cell surface after they have been bound to LDL molecules. LDL molecules containing cholesterol are broken down, and the cholesterol molecules are passed on to either the lysosome or the endoplasmic reticulum for esterification. In this diagram, we look at the differences between a regular individual's cholesterol intake and an individual suffering from Niemann-Pick type C. Type A shows endosomal transport in an individual who is not affected with Niemann-Pick disease. In this diagram, it is observed that the unesterified cholesterol enters the early endosome, where it passes on to the late endosome. Here, unesterified cholesterol molecules are passed either to the lysosome or the endoplasmic reticulum. If it goes directly to the endoplasmic reticulum, it is esterified and then passed to the Golgi apparatus, where it is sorted and disseminated throughout the cell. If, however, it goes in the lysosome, the unesterified cholesterol goes firstly to the Golgi apparatus and then to the endoplasmic reticulum, where it is esterified and passed back to the Golgi apparatus, which disseminates the esterified cholesterol. However, in an individual suffering from Niemann-Pick type C, the NPC1 and NPC2 genes are defective. In these individuals, the Rab7 and Rab9 protein, not shown here, but which are usually present on the late endosome, which binds to either the lysosome or endoplasmic reticulum, is defective. What this means is that the late endosome cannot, as is seen here, bind with either the endoplasmic reticulum or the Golgi apparatus. This results in a buildup of unesterified cholesterol within the late endosome and the lysosome, resulting in the enlargement of cells, in which this gene deficiency affects most.